The following was produced by researchers at the University of Georgia in the Warnell School of Forestry and Natural Resources. This demonstration video describes the harvesting of xylem and phloem tissue and subsequent RNA isolation from Pinus tita, commonly known as Loblolly Pine, which is the single most important conifer species for forest products production in the United States. Once the tree is felled, in this case a 40-year-old tree, the main stem is cut into bolts that are approximately 50 centimeters in length. The bolt is then scored longitudinally with a chainsaw in several places to facilitate removal of the bark. Using a hammer and wood chisel, the bark is lifted up until it can be peeled away in sections. Next, the phloem tissue is removed by hand and is stripped from the inside of the bark sections and immediately immersed into liquid nitrogen. Once the bark has been removed, the thin layer of secondary xylem can be collected by scraping the bolt surface with a single-sided razor blade. The watery xylem tissue is immediately placed into liquid nitrogen. In this harvest, we also collected samples of reaction wood, commonly referred to as compression and opposite wood. Before cutting limbs from the main stem, the limb orientation is first marked with spray paint so that the areas containing the two tissue types, opposite wood on top of the limb and compression wood on the bottom, can be determined during bark removal. The limbs are first scored with a chainsaw along lines delineating compression and opposite wood, and bark removal is performed as with the main stem bolts, except that only bark in the region corresponding to the type of reaction wood being harvested is removed at any given time. It is typically more difficult to remove large bark sections from these smaller limbs. Secondary xylem reaction wood samples are collected in the same manner as with normal secondary xylem by scraping with a razor blade and immediate immersion into liquid nitrogen. Note the reddish-brown color characteristic of compression wood. It is noteworthy to mention that collecting and processing multiple samples from a large tree requires many hands, with each person involved performing a designated task so that all tissue types are being collected simultaneously to help reduce RNA degradation. Samples frozen in liquid nitrogen are subsequently removed to labeled storage bags and placed on dry ice for transport back to the laboratory. Our first step is to pulverize the tissue using a Specs Model 6850 freezer mill. Compared to grinding with a mortar and pestle, this step significantly increases RNA extraction yields for woody tissue samples. Keeping the samples cold with liquid nitrogen, the tissue is first broken into smaller pieces by hand and placed into the sample chamber. The chamber is inserted into the carrier, locked in place, and the sample is milled for 2 minutes. The final product has the consistency of a coarse powder. The RNA extraction protocol is a 2-day process. On the first day, 2.5 grams of frozen tissue is added to RNA extraction buffer that has been heated to 65 degrees Celsius. The sample is shaken vigorously to mix, and an equal volume of chloroform is added, and the sample is mixed again with gentle inversion. The sample is next placed on a rotating wheel for 5 minutes. The sample is then transferred to high-speed Teflon Oak Ridge tubes and centrifuged in an SS34 rotor at 12,000 RPM for 5 minutes. The upper aqueous fraction is extracted into a fresh tube, and a half volume of chloroform is added. The sample is vortexed for 1 minute and again placed on the rotating wheel for 5 minutes. After a second centrifugation, the aqueous fraction is again extracted into a 50 ml high-speed polypropylene tube and centrifuged at 10,000 RPM for 20 minutes to remove any residual particulates. The sample is decanted into a 50 ml Falcon tube, and a 1/4 volume of 10 molar lithium chloride is added to the sample. The sample is mixed by shaking and placed at 4 degrees Celsius overnight. On the second day, the lithium chloride precipitated sample is first centrifuged at 11,000 RPM for 30 minutes at 4 degrees Celsius. The supernatant is poured off and discarded, and the tubes are inverted onto chem wipes for 1 minute to remove any excess liquid. The RNA pellet, which should be visible, is resuspended in 800 microliters of hot SSTE buffer that has been preheated to 65 degrees Celsius. Be sure to pipette up and down several times and all around the pellet margins to ensure that it all gets into solution. Next, transfer the resuspended RNA into a 2 ml Ambion RNase-free microfuge tube. Heat the sample for 1 to 2 minutes in a 65 degrees Celsius water bath, followed by vortexing to ensure the sample is completely resuspended. This can be repeated if necessary. Now add an equal volume of buffered phenol chloroform. Vortex the sample for 15 to 20 seconds and microfuge at 14,000 RPM for 3 minutes. The aqueous fraction is extracted into a new microfuge tube and an equal volume of chloroform is added. The sample is vortexed and microfuged as in the previous extraction. For the final organic extraction, phase lock gel tubes are first microfuged at 11,000 RPM for 2 minutes. Next, the RNA sample from the previous extraction is added to the phase lock gel, followed by a half volume of chloroform, and the two are mixed by pipetting gently. The sample is then microfuged at 11,000 RPM for 5 minutes, and the aqueous RNA sample is transferred to a new Ambion RNase-free microfuge tube. The next step is a simple RNA precipitation. To the sample, add a 1/10 volume of 3 molar sodium acetate, pH 4.8, and 2 volumes of 100% ethanol. The sample is vortexed briefly, placed in a -80 Celsius freezer for 30 minutes, and then microfuged for 30 minutes at 4 degrees Celsius. The supernatant is carefully aspirated, and the small white pellet is washed with 1 ml of ice-cold 70% ethanol. Aspirate and repeat the ethanol wash. After the final aspiration, residual ethanol is allowed to evaporate by leaving the tubes open on the bench for 5 minutes. RNA pellets are resuspended in 100 microliters of DEPC water and resuspended by vortexing and heating at 65 degrees. This can be repeated if necessary. Sample concentration is determined spectrophotometrically, and 262/80 and 262/30 ratios are calculated to estimate the extent of protein and carbohydrate contamination in the sample. 1% agarose gel electrophoresis is used to qualitatively estimate sample quality, genomic DNA contamination, and RNA degradation. Sample integrity of critical samples is evaluated further using an Agilent 2100 bioanalyzer to determine ribosomal 28-18S ratios and an RNA integrity number. Thank you for watching our video on the harvesting of tissue samples and the isolation of total RNA from Loblolly Pine. The following individuals were responsible for the filming and production of this training video.